Hello, welcome to this next episode of the Progressive MS webinar series, where we're trying to make you a little bit more resilient in the face of a progressive illness. Today, we are talking all about research, and I'm delighted that we have Dr. Una Fitzgerald with us today, who is from the University in Galway. Dr. Fitzgerald's research focuses on developing an understanding of the molecular mechanisms underlying neurodegenerative disorders such as multiple sclerosis and Parkinson's disease. After graduating from NUI Galway with a BE Industrial in 1983 and an MSc in Biotechnology in 1985, she worked in the biotech sector for 5 years before returning to academia to complete a PhD in molecular biology in the University of Strathclyde, Glasgow. Dr. Fitzgerald returned to Ireland in 2003, taking up a lectureship role in biomedical engineering at NUIG in 2005. Since then, the main focus of the Fitzgerald Group has been multiple sclerosis and other brain disorders, including Parkinson's disease. The group has also studied the normal development of the cerebellum. Dr. Fitzgerald was the director of the Galway Neuroscience Department, and she represented Ireland on the MS International Federation's International Medical and Scientific Board. She has also recently joined the committee of the Dementia and Neurodegeneration Network Ireland. Since May 2019, she is coordinating a 3.9 million EU-funded project that aims to work with researchers from Denmark, Germany, Belgium, France, Italy, Spain, and the Czech Republic to create new devices and treatments for multiple sclerosis. Very welcome, Una. Thank you very much for joining us.
Thank you so much, Robert, for inviting me to be here today. I'm absolutely thrilled and really looking forward to getting into the whole research thing, for MS. So really, really happy to be here. Thank you very much.
And I think it's really important that we understand what is happening at the moment and how things are perhaps going to develop into the future. Mm-hmm. But reading your CV or reading that out, I'm wondering, engineering and now MS.
Yeah.
How did you get here?
Well, I can blame two older siblings for doing engineering, actually. And I also loved maths when I was in school. So it seemed like a natural thing to do. Having said that, one of the siblings became a surgeon, so we kind of have a bit of a habit in our family of kind of ducking around through different disciplines. But after doing the engineering degree, which I did enjoy, and being in the biotech sector, I was noticing that lots of questions would come up, because when I was in that company called Celtech in Slough in England for a few years, we were making EPO, which people might have heard of, erythropoietin. So that's a very popular drug on the black market, actually, but it is.
And what does that do?
It boosts the amount of red cells in the blood.
Ah, okay. Right? This is the EPO doping from.
Yeah, exactly, exactly. So we set up a whole manufacturing facility to make that. But I couldn't help but notice that the people who were based in R&D seemed to be having much more kind of fun, and while I was based on the other side of the fence in manufacturing, I couldn't answer a lot of the questions that were coming up, like, why did we only get 4 grams of EPO in this batch, or 3 grams instead of 4 grams in our EPO batch? And it was all about needing to understand this live system that was being used to make EPO, so when you keep asking questions like that, it means that probably you should be doing research, So I made the decision to leave industry and go on. Actually, it was an industry-linked fellowship scheme that existed in the UK, and that allowed me to get funding for research at Strathclyde University. So yeah, that's how I got into research, and I was working on creating permanent cell lines that were generated from the human umbilical cord. So that was pretty interesting for me.
Is that kind of connected to stem cells?
It wasn't at that time. We didn't even know about those stem cells. No one knew about them. And so my PhD quite a while ago. So, but at that time, we'd never heard of stem cells from the umbilical cord. So if I'd known.
So you were creating cells
from the umbilical. So we were using, it was at that time, the technology that was used again at that time was to take what's called an oncogene. So in other words, a gene that's known to be associated with cancer, and then artificially put it into that endothelial cell. So that's a cell that lines the blood vessels within the umbilical cord, and make it behave like a cancer cell, i.e. be able to grow endlessly. Well, it was immortalized is the way, that's the language that's used, you know. So yeah, it was pretty interesting. And then following on from that, I had gotten into, I'd got a job after that in a cancer research institute called the Beatson Institute in Glasgow, which I enjoyed very much. But, you know, parallel to all of that, I did have an interest in neuroscience. That was always there in the background.
And is there a reason that it was in the background?
Yeah, there is a reason. It's a nice reason, because my father used to be the professor of neuroscience professor of anatomy at NUI Galway, but his specialism was neuroanatomy. Oh. And so he was pretty obsessive about this, and he's written a textbook on it, which is now called Fitzgerald's Neuroanatomy, or Clinical Neuroanatomy and Neuroscience. And it's in its 7th edition. But basically, we grew up in our family with my dad, like if I did this, you know, lifted up a cup, he'd go, oh, you've no idea, you know, what's involved, what had to happen for you to be able to pick up that cup and hold it there and look at it and all that sort of thing. So this was, you know, that was constantly in my ear, you could say. So when an opportunity came up after the cancer research postdoc, basically down the corridor from where I was based in, and it was Professor Sue Barnett's lab. So some people may have heard of Sue Barnett. You know, I went for it, and I have not looked back since. And so while I was in that lab, I was delighted to be learning for the first time about the oligodendrocyte in great detail. So
that oligoclonal banding, and I remember when I heard,
yeah, it gets confusing. Yeah.
I remember when I first heard that when I was diagnosed, oligoclonal banding, and I had absolutely no idea what that meant.
I can understand why, you know, because it was based on a Greek word, probably. Is it oligo, like is Greek? And clonal. Yeah. Well, you see, the thing is, it is confusing within the MS field, because the oligodendrocyte doesn't really have anything to do with oligoclonal bands. They're two different things completely. Oligoclonal bands are generated by immune cells. Okay. You know, within the CSF, the cerebrospinal fluid. Oligodendrocytes are the brain cells that produce myelin, which is the thing that's under attack during MS.
So what's the connection between the two?
There isn't any, apart from the fact that, you know, the oligoclonal bands will demonstrate that immune cells are functioning abnormally. Okay. That they are producing antibodies that are, you know, probably attacking your myelin. You know, so there's no direct relationship, is the way I would put it. You know, it's the oligoclonal bands demonstrate that disease is there, and it's your kind of go-to way of well, it's one of the criteria used in the diagnosis of MS.
So that's your biomarker that people are always looking to have something that says this is definite that you have. But I presume that banding occurs in other illnesses as well.
I'm not sure. I wouldn't like to. I don't know. Maybe. Yeah. I haven't checked, but it's a very good question. So, but yes, I imagine other autoimmune, not necessarily autoimmune, but infection-linked disorders. So you're probably right. Okay. Yeah.
Just to let you know, we have a question and answer function on this for all the people who are
fantastic.
Who are listening. So if they want to ask a question, or if there's something on your mind, we will see if we can address it. And if we can, we will during the course of this. But it all depends on Una's actually expertise. She's got a particular area that she's interested in, and we were going to discuss what you're doing in Galway. Also, Aoife, if you wouldn't mind putting up the poll, so that we can see what type of people are actually attending this. So while we're talking, people can actually answer that poll. Okay. I'm just wondering, you know, given your father's thing about neurological, and you went into engineering, did you find that the work that you did or what you learned in engineering prepared you better for the understanding of the neurological system?
I think that the engineering gave me a certain mindset that I had when I went into research, because, and the funny thing is, which we can get back into in a while when I talk about our current research, because I've kind of circled around again and I'm back in the College of Engineering, but within the biomedical engineering discipline. But I think it gave me a mindset when I went into research during my PhD, which didn't really fit with the mindset of a researcher, because engineering is very solution oriented, in my opinion. So an engineer will go in and look at a problem and say, okay, how can we fix this? And they'll want to see the solution, you know, within a year or whatever. That's kind of how I see engineers. It's very practical and applied, and they apply their skills in whatever discipline within engineering. And so it meant that it took me a while to be able to converse with research scientists, you know, in the kind of language that they use. Because if like an engineer would say, well, what about this? Why aren't we doing this? And they go, well, you know, that depends. You know, it depends on the cell type. It depends on the condition. It depends on the age of the person when the sample was taken. It depends on exactly the question you're asking and exactly the methods you're using and the day you're using them and how long you're using them for. And, you know, basically it's not that simple, you know? So I appreciated, yes, it gave me skills. I was probably more organized than the average PhD student coming in, but I was 28 years old when I started my PhD, you know? So I was a mature student, a little bit more organized, but I didn't have that research. I didn't quite, it's built up now, it's better, you know, but I kind of went in with a different mindset. But, you know, interdisciplinarity is extremely important now. It probably always has been, but it's where a lot of interesting discoveries are made. So a mixture of backgrounds is very important, I think.
And now leading into your own research that you're doing in the university, what particular areas, now from my understanding of research into multiple sclerosis, there are three kind of areas. One is kind of prevention, one is repair to an extent, and the other one is preventing that damage from happening. Do you know?
Yeah.
So
I understand what you're saying. It's probably, that's a perfectly good way of looking at the whole sector or the whole sphere of research, I think, from the point of view, especially of someone with MS. So I think it's really interesting to me, actually, that you've expressed it that way, because we might rewrite some of our narratives, you know, but it's just, I would probably say the third one that you mentioned there was, what was the third one? I can't remember that you said, because I've a feeling that a lot of what we do fits in there in your third option.
It's kind of
but nonetheless, I think that basically the way I would say from a researcher's perspective, another way, so it's a bit different, another way of describing the kind of research that we do is to start by saying, well, someone like me or the people in my group probably mostly could be described as doing basic scientific research, or bench research it's called, because we're literally at the bench in the lab doing our research most of the time. And there would be people from so many different disciplines doing bench research, the biochemists, even microbiologists, because that's to do with, playing around with gene sequences and things like that. We have the physiologists, the pharmacologists, and then bioengineers. So that's for the kind of basic bench type research. But then, and all of those coming together is really, really important. The chemists, of course, let's not forget the chemists. So then another kind of research, which we do want to get into, people call translational.
Okay.
Right.
Can you
I
explain
don't know if you've
that?
heard, have you? Yeah. So this is a term that's everywhere on grants, written by people like me to funding organizations. This is translational research. What does that mean? It means that this idea will be translated into the clinic. It's not like it's translated into another language. It means it will translate. This idea can be brought in to the clinic. And that's the kind of thing you need to say or imagine or envisage, that this is where my research is going. And it makes it more likely that you will get funding, obviously, because those funding organizations are trying to improve the lives of people, for example, with MS, So translational is one. But that kind of encompasses a lot of different kinds of clinical research, which I think overlap with what you said. Do you know what I mean? So the kind of prevention, I'm not in that space at all.
Okay.
You know, not yet, at any rate, you know. But again, I'm going, how would I do that? You know, the kind of the day-to-day, the research that's around day-to-day kind of research, day-to-day lives of people with MS would be the physiotherapy research, occupational therapy research, psychology, you know, with obviously the physiotherapy, like work being done by Susan Coote right now, exercise research being done by Eric Downer, for instance. That's research. You know, it's a different kind of research. It's not the research I'm doing. But as I might explain in a minute, we would like to work more closely with people who are recruiting, who are already recruiting, or who plan to recruit people with MS and others for comparison in their future research studies, so that we can access some samples basically from these people.
So like, for example, I work on the COB MS trial, which we're currently doing, which is a new cognitive therapy for people with MS. So you're looking for the people that we recruit, say in our trial, that they would also, or the information that we have can also be used for your trial. And I remember actually reading about this and actually attended a webinar about it, the importance of all research in a particular disease area, answering one or two of this two questions. Everybody has to answer these two questions, and then you answer sub questions, and creating a list of, well, we have to know about this, this, and this. These are our priorities. Is that what you're trying to create, an environment where research that's happening here in Ireland is following a general theme to tackle specific questions?
Yes, I would say it's a mixture of things, because, you know, what I didn't manage to explain there early on is that if you want to have a career in research, and let's say it's MS research, individuals at the early stage of their career actually have to find their own niche. You know, what is it that I'm doing that separates me from anyone else who's going after the same money, essentially? You know, because you can't actually do your research unless you are supported, because it's an expensive endeavor, really. So in the beginning, you have to find your own niche. And, you know, Professor Denise Fitzgerald, for instance, in Queen's, you know, her niche is, I know she does lots of things, but one of them is T-cell biology. You know, Claire McCoy in the RCSI, her niche is microRNA. She's doing fantastic work on that. In the beginning, I worked on a stress signaling pathway. Now, and I don't mean like cortisol or anything like that stress. People might understand that stress. This is a sort of a subcellular stress that impacts just one of the kind of regions of this within a cell. And it's called the endoplasmic reticulum. And we spent years, in fact, we were the first to do this in the context of MS. So you find your niche, but like stepping back, each of us, as we've done, stepping back now that we've spent, I don't know, 20 years on MS, you recognize from going to conferences and attending more clinical meetings that we need to connect all of this together. And that, I mean, look at what's happening. We have failed to produce enough decent treatments for the later progressive phase of MS, right? And everyone acknowledges that now. But the detail that we need to extract from our studies is so complicated and so extensive and so high volume that we have to collaborate. We have to have the engineers, we have to have computer scientists, data scientists, we have to have statisticians, we have to have, you know, the lowly bench scientists, you know, who might have the idea in the beginning, but it has to go through all of those kind of hands. The clinical experts, the neurologists, we have to have the MS nurses involved to help us recruit people, and then others who are doing other kinds of studies within those other fields like physiotherapy and occupational therapy. So I don't think we're all, our common goal would be to improve the lives of people with progressive MS in particular. So that's a common goal, and it's a common question for all of us. How are we going to improve the lives of people with progressive MS? What approaches can be used? And so to study it from the inside out, so looking at tissue from people with MS. Outside, okay, well, I know, we can't really experiment on your brain right now, so can we just take some samples and see if those samples have bear any relationship to what's going on with you now, today?
And is that what you're doing at the moment? Are you just,
well,
so you're looking for a biobank of?
Okay, yeah, that's a good question. So there's a couple of things. I could get into a couple of big projects that I'm starting or have started and then will be starting if it's a good time to do that now. Yeah. Okay. So since you mentioned in the intro that we have a large EU project that's funded by the EU, and it's one of these big sort of training networks. It's very important for training the next generation of young researchers and who will become older MS researchers. So the goal with that project is to design a new kind of device, medical device, that would be used that would be targeting the sort of outer areas, the outer rim, let's say, of the brain. Because this particular part of the brain, in fact, is associated with cognitive deficits within MS, but also possibly if it's in the kind of what's called the motor cortex, it could be affecting how people are controlling their movements. So we're trying to see if we can just focus on that pathology, the one that's just in the cortex, understand what's going on there within the brains of people with MS who've had these kinds of, who've had myelin removed from that part of the brain because of the disease. And then say, well, okay, we've got a picture now of what's happening there. And many others have studied this area as well. It's studied by a lot of people, and much larger groups than ours actually. But and we've taken that information to inform what we're doing. And our question is, well, can we, using this medical device that contains a biomaterial? A biomaterial just means it's like a gel is one way of looking at it, right? So let's say we're putting a gel inside a device and we're using it to control the release of, firstly, an anti-inflammatory compound, and then secondly, something that might boost repair. So in other words, the first release would be for creating the conditions that would allow the repair to happen. Because if you just take let's say not a regular, but let's pick any MS lesion, because of the damage that's present there in the lesion, it's blocking any repair. So we know there are molecules there that just prevent repair happening. Even though there are other cells trying to get in there, you could say, but they're being inhibited. So if we could do something about creating better conditions for the repair to happen, great, And we also understand that the people with MS, or we believe that they don't want something that has to be administered every week, every day, if possible, avoid that. So hence the idea to use the biomaterials to help control that release. And the reason that we were able to do this is because we are based in Cuarim, which is the Irish word for care, which is the National Science Foundation Ireland funded Centre for Research on Medical Devices. And I think we're very fortunate in Galway to be based within Cuarim. It's a big lab, 60 researchers at kind of all levels. So our group is sitting within there. And we're surrounded by chemists, bioengineers, and people with a whole range of expertise who already had that knowledge about how do we build this scaffold? How do you use it to release drugs? What are the conditions? What should it look like? And all of that is dependent on which drug is picked. So different members of this consortium, in the other countries, in Denmark, in Germany, in Belgium, every researcher has their favorite compound, That's the other thing. You've got your niche, but you've also got your molecule, and it's my molecule, I want to work on that, So, but because with this project, we are forced to pick two, that's the concept. We're forced to, in the end, try and pick two or have a consensus. So we're getting there. And what's happening in the different locations is those labs are using their expertise to demonstrate or to test a whole range of compounds. and say, well, actually, this one looks good, because we have a molecular weight cutoff as well. So it can't be massive molecule.
Okay.
So that kind of that feeds into that decision. So that's the big EU project.
Mm-hmm.
And the second thing I'd like to bring up is a couple of new projects that are going to be starting from about September, October.
Okay.
And this is funded, the two of them are actually funded through another big doctoral training big doctoral training network that's funded from the UK. I think it's EPSRC and Science Foundation Ireland, again, through CURM.
Okay.
And one of the projects, now this might blow your mind, right? Because it's blowing my mind, I can tell you that. It's taken me 6 months to get a handle on it. But and in fact, it took me a month to even write up the project description. Mm-hmm. So it's using a technology called organ on a chip. I don't know if you've heard of it.
No.
I'm not surprised, because it's very kind of current, it's very kind of now. And I would say others, of course, in the US and in bigger labs outside Ireland are already on this, okay? But nobody has produced what we call MS on a chip, all right? And that's, now what that means, in fact, is a fancy name for a more advanced lab model of disease, okay? So it means that effectively what we're doing is we're miniaturizing existing lab-based culture systems. So it's literally taking something, you know, that might be this size, the size of your phone, right? And so I might, let's say this is a cell culture flask. If I was at work, I would have a cell culture flask, but I'm not allowed in until the 20th of September. So if this was a cell culture flask, I might have my oligodendrocytes cultured in here. Mm-hmm. And they're, you know, it's all going well. They look good. They're showing the properties that I want them to show. Mm-hmm. But if you can imagine now miniaturizing that so it's almost the size of, you know, the little corner here.
Yeah.
Okay. The advantage of doing something like that is, and we use a thing called microfluidics, you know, which again, you know, it's
very technical.
Jargon. Yeah, it's jargon. It just means it's small. Okay. You know, and there's fluid in it.
Okay. All right.
But the great thing about it is what has been designed by engineers, I have to say, mm-hmm. and that's why the engineers are really important, and the physicists. Let's not forget the physicists, because they're doing incredible work on sensing technologies. But the physicists and the engineers here are very important, because when you miniaturize mm-hmm. your culture systems, they're very hard to see for a start. And if you want to do sort of real-time monitoring of everything, you know, you want to have nice sensors in here. Okay. And then the other thing that the microfluidic technology does, though, is it allows you to connect. Let's say I've got oligodendrocytes here in this little chamber, mm-hmm. and over here, halfway along this microfluidic device, I've got my immune cells.
Okay.
You know, and in particular, one of the students on the PMS Mat train is working on the B cells, I'm sure you've heard of. So they're the ones making antibodies. Mm-hmm. And in particular, in the cortex, lots of huge evidence, you know, from Richard Reynolds and others, Professor Richard Reynolds in Imperial College London, showing that these B cells gather in the folds of the brain mm-hmm. for some people with MS, not everybody. Okay. And they might gather there. So they're not in the brain. They're not, you know, themselves going into the brain tissue. They're just sitting there in the folds. Yeah. They gather and they start to secrete toxins that just go, hey, let's destroy this myelin, you know. Okay. And so she's trying to model that. But then I have another student who's looking at different kinds of neurons, of which there are like 13 or something just in the cortex, saying, well, which one of these is the thing that dies? Which neuron is the one?
They don't know that yet already.
Pardon?
They don't know that already.
Well, you know, it's only recently, and that's through work in, well, I think in others, but Anna Williams, she's a neurologist and a researcher at Professor Anna Williams at Edinburgh University. And there's some hints now at which particular one we should be looking at, which is helpful because we really don't want to have to look at 13 different kinds of neurons mm-hmm. if we can possibly avoid it. So our student Daniela Costa is working on those cortical neurons, and Gosha Dombrowska is working on the B cells in the toxic niche. So we're very happy to have that happening. And then there are other researchers that are being supervised by the director of Quorum, Professor Abhay Pandit. And one of them is looking at signals from sugar molecules on the surface of cells.
Okay,
Can
I just pause for 1 second? I'd like to close the poll, Aoife, if we can.
Yeah.
And would you believe it, Una, we only have 15 minutes left. So
we're okay, I better get on to some other stuff.
Yeah, that's it. I'm just, because we could listen to you for, there's so much stuff. So I'm just going to read the poll here just for 1 second. We have, the majority of people are from Connacht and Leinster, but we do have some from overseas, UK and outside of USA and the UK.
Brilliant.
It's almost an even mix of people between primary, secondary and relapsing remitting MS. Okay. And we have small family members. The primary age group is from 46 up, with 52% 60 plus, right, attending. Okay. So that's actually very interesting. The age profile, considering that you're interested mainly in the more progressive or the more advanced form of MS, it's kind of your right audience for this. So sorry, you're about to go on to the third.
Yeah, but what I would say, yes, our focus is progressive MS, primary and secondary. But I would like to say to the others with relapsing remitting that anything we do is relevant to that as well. And anyway, in a study, we would always compare relapsing remitting with progressive, so they're not left out of, it's not like we're not thinking about relapsing remitting,
Just so as a side note, do you agree with those three categories? do you think MS is just a spectrum and it's all just MS, and that defining it as these three separate types is that actually beneficial to research, or does it limit you?
It doesn't limit me, because as I said before, we have to make a decision on where we're focusing, and kind of once we make that decision, oh God, my neighbor is ringing the doorbell. I should have told him not. He's only 5 years old, so I'm going to try and ignore him.
Okay.
So yeah, like we pick the thing we're focusing on. Okay. Do you know what I mean? So to us, those definitions don't matter too much, because we're so deep into the underlying mechanisms, in a way. Yes. In a way. And we could say, well, whatever we discover, it's either going to be applicable to one or the other, or all of them. Do you see what I mean? So it's kind of, I don't know how important it is. I mean, the fact is 80% of people have a relapsing remitting course. So I don't think that's going to change. Yeah. You know what I mean? After that, it might get complicated, and you've confounding pathologies because you're older, or you've other conditions, so it gets very complicated and more difficult to study for that reason, and to research. But the second thing I wanted to mention, though, and I think it'll bring in some of the other things you might want to learn about, is a project that we call the point of care device for people with MS. And this, I would say, is more translational. Okay. I'm hoping it's more translational, right? Yeah. Because it's called a point of care device because that's exactly what we wanted to do. So it's like, again, it might be, a phone-sized gadget is one way to describe it. And this work is going to be done in collaboration with Dr. Sinead Hines at NUIG, and who you know very well.
I do.
And we're delighted, because she's literally across the building, across the road from us at NUIG. So it's very, very convenient. And Sinead has been fantastic on advising us on this upcoming project. And one of the students, Bianca Castelli, is one of the students who's going to work on this. Another one called Shima Shipuri, who's funded through a Hardeman scheme, so not that doctoral training one. They will both be working on this point of care device idea. Why Sinead is involved, it's because, can you hear that doorbell?
We can, yes. This is real life, Una, and I wouldn't worry about it too much.
Okay. So the point, so what was I going to say? Yeah, because I've kind of lost my thread there.
So this point of use device. So
why Sinead? Why Sinead is involved is because of her work on. Sorry, I'm at work. Oh, he's sorry about that. Is that she is working on cognition, right?
Okay.
And of course, as I said earlier, it's the pathology in the cortex that's partly contributing, at least, to cognitive deficits in people with progressive MS. So it seems logical that we should try and learn from each other. And exactly, it's a good example of that thing of multidisciplinarity, you know. And so we started talking to Sinead, and she helped with the applications for funding for these students, which were successful. And now we're going to collaborate with Sinead on, let's say, okay, and this is a call out now to people, because we want to recruit people for this. And more importantly, we want to get people involved before we actually do the study. And I know we might get to that in a minute, but so anyone who's interested in this kind of research, please do get in touch with us. But the idea is that, let's say someone will, you know, we've recruited somebody, they meet the criteria, you know, they arrive, they meet us in whatever platform, maybe it's online initially, or they attend in person, and they might have just done the cognitive test that Sinead runs. But we also learned of a study done about smell discrimination that was published, and it was, okay, it's one paper, right? But on a small number of patients, but they showed that actually smell discrimination can separate the progressive MS cohort from the others. So that just sort of put up a flag, or flashed a light and said, that's interesting. Because I had a previous study, the one who was working on Parkinson's, and he was doing smell testing on Parkinson's, you know, or for a Parkinson's study. So we thought, oh, well, we know the smell test kit, or the smell test thing, so we'll just bring it into the MS thing and add it on as a functional readout. So here we've got the person, we've got their smell, we've got their cognition, but now in the same period, ideally the same day, the same time, even the same hour, we asked them to spit in a tube. And like the COVID test, can we grab a bit of your nose sample? You know, at least you've been through it, so you know what it's like. But when it becomes a point of care thing, it means that we also want to, like the COVID test or pregnancy test, you want to pop that sample, you know, into the device and get an answer there and then. Do you know what I mean? But what we'd like to do is resample them an hour later or this evening, ideally. So maybe it'll have to be modified in some way that the people take the samples themselves, or you know, there'll be a bit of training back and forth. So we really need the help of the MS community to do this, I think. So, but I'm really happy. We have 2 students on this. They'll be working on different aspects of it, and you know, I hope that we can collaborate with so you.
You gave us links to those earlier today, and Aoife will put them in the chat for everybody to access. And when this webinar goes onto YouTube and it's on our podcast, we'll also have the links there as well. Okay. I'm just, so do you think that test is going to be a diagnostic test for MS, or is it helping you with what part of the spectrum you're on, so that you can then tailor your treatment?
I think it's more the second. So we're really more, our hope, our ambition would be that it would be a tool, it'd be part of the tool that the neurologist or the treating person is going to use to decide what to do. You know, or maybe you can give a richer view, like an internal view of the biology that happens when someone follows an exercise regime or does one of the occupational therapy things, you know, or maybe there is a response, but it's temporary, or how long does it last? But the only way we'll get that is if we do this kind of sampling, this kind of biomarker sampling. I mean, the most popular way of doing biomarker studies is using MRI, but it's expensive, you know, it takes time, there's a long delay. So it's in that kind of in-between space, but I think it's more about monitoring ongoing.
Because I'm just after going through a course of IV steroids, and they've had phenomenal benefit for me. Really phenomenal. I was barely walking, and now I can climb up the stairs.
I've heard that sort of thing.
You know, and it is, in one sense, it's almost miraculous. But it would be interesting then, though, in the midst of that treatment, just before that treatment, doing this test, seeing what kind of story that tells, and then when I was starting to walk properly, then you can say, oh, well, this has disappeared, or this is appeared in the test. Is that the intent?
Yeah, it is the intent, because yes, I mean, the logic would be not only would we inform the clinician potentially on which, if someone's coming off one treatment and going on another, because there's so much research being done even on that. How should they come off? What dose should they get? What dose should they use when they're going back on it? This sort of thing maybe could help, especially if it's quick and easy, so that's probably what I would hope it would be used for. Yeah. And then afterwards, like you say, post-treatment, or what's happening there, how long is it lasting, what's changing, but the decisions around the markers are difficult. Are they meaningful? How do you demonstrate they're meaningful? Which things should we look at? How many assays can we have in this in this point of care device? And we're collaborating with Professor Jonathan Cooper in Glasgow Uni on that. But the other thing I did want to mention is our All Ireland MS Research Network before we finish, if that's possible.
Yes, absolutely.
Yeah, because you'd mentioned, do you remember I was saying about multidisciplinarity?
Yes.
And also this idea that people, more experienced people within the field, like myself, and I'm I'm a lot older than Denise, but Denise has been in the field for a long time, a T-cell biologist, Denise Fitzgerald, and basically stepping back from it and saying, well, now we've got quite a few people on the island of Ireland who are working on MS. Clearly, Susan Coote in University of Limerick, and Eric Danner on exercise in TCD, and Kumlesh Dev in TCD, Kingston Mills, of course, in TCD. And then Claire in RCSI, and Valeria Gutzman, also at Queen's University, an independent fellow, who very interestingly is working on aging oligodendrocytes. So imagine aging has an effect on lots of things, but she has a particular interest in how is aging affecting the ability of oligodendrocytes to repair lesions, So all of that stuff is really good, but we realize that, well, actually, each of us have our own little areas, but if we collaborated, and if, let's say, the tech that's available and the expertise in Quorum, for instance, is exploited, because we could maybe play around with candidate compounds and see how they can be released, and think about where devices can be placed, or all of those point of care devices. We really need to come together as a research network. So we worked for a year together. So that was Professor Denise Fitzgerald who proposed the idea in the first place. And then with her, Yvonne Dombrowski, who is a big name in the inflammasome field. Okay, so Yvonne is a fantastic researcher. So Yvonne was the first author, and Denise was the last author on a Nature Neuroscience paper. And so they're really top research scientists. And Claire also incredibly successful in the area of microRNAs in MS. So we got together, and Jill McMahon in my group, who has decades of experience of looking at postmortem human MS tissue. So I was delighted when I was able to recruit her into my group. She's an expert on gene therapy as well, and on stem cell biology. And I think you might hear from her in a later episode. So, and then I'm delighted that Aoife Kerwin is now on our committee for the AIMS-RN, making a huge contribution, especially around social media and tweeting what we're doing, and giving us the perspective, like yourself, Robert, from the perspective of someone who has MS. So really, really delighted that Aoife is involved. But we set up this network. We took a year to do it, to get the website up, to get the branding done, funded by Quorum, I have to say. The graphic artist, Maciek Docik, fantastic job of branding. And we launched at the end of February, and we had our first successful research scholarship, studentship funder, students who were funded for the summer by generous donation from Eamon Houghton and Declan Smith in Chemical Systems Control, plus a private donation through our foundation office at NUIG. And just if anyone's interested in helping with that sort of thing, it's about 2,000 per summer student, 2,000 euros. It's not a massive amount of money, but incredibly important to those students. So we had like something like 70 applications for those 4 studentships.
Wow.
70.
Wow.
I mean, like, what does that say?
Huge interest in this as an area.
Yeah, and about half of the applicants had a reason, like a personal reason.
Okay.
They either had a family member, a friend, So it's hugely important on the island. And I think the bringing together of what's there in Northern Ireland with Denise, Yvonne and Laria, with everyone else who's in the Republic. We need to collaborate more, and we hope that this network is the thing that's going to make that happen, you know.
So, you know, Una, would you believe it? 45 minutes has already passed.
Oh God.
Right.
Okay. I wouldn't believe it.
And we have, I think, barely scratched the surface.
Yeah, I would say that's true.
There is an awful lot more that I'm sure that you can tell us. And
yeah.
you know, things that I would love to learn about, you know, neuroplasticity and about, you know, using existing medicines, repurposing them.
Yes.
Things like, and I use cannabis myself as a treatment for spasticity, and the impact that that has. And I know
you should interview Eric for that.
and that's another plan of mine. So I have to thank you so much for your time today, Una.
Not at all.
You know, I would love to go, maybe we could have another opportunity to have a discussion about what's going on, and maybe make this more of a regular thing where we can share what's happening in research throughout Ireland, but also around the world. Just to let everybody know, the next episode is going to be on September 30th, and we're going to be talking about relationships and multiple sclerosis with Dr. Jonathan Egan. Yeah.
I did want to make sure I mentioned that MS Ireland was critical in my early funding. You know, they were the first people to give me funding for my MS related research when I came back to Galway. Really, really important.
And this shows you the power of the network. It shows you the power of the community.
Yeah.
And it shows you that we actually, as people with MS, have a role in research, the direction of research. And now we've got an invitation from Una to take part in ongoing research. So I have to thank you very much for that. And we will talk again.
Delighted. Yeah.
I again have to thank Aoife Kerwin and Aidan Larkin for making this webinar series possible, and for MS Ireland for supporting it. I think it is truly invaluable to people of all types of MS, whether you're a family member or somebody with the illness. So thank you very much, and goodbye, everyone.
Thanks a lot. I really enjoyed it. Thanks very much.
Thank you. Bye now.
Bye.